Hello, guys. Welcome to second session of protozoa. We already talked about the characteristics of protozoa, and then sorry, I don't need it, but that's okay. And then this is the second section of protozoa. Then the very first phylum I talked about it was Euglenozoa, and then there are two organisms in this phylum that are significant. So they are Trypanosoma and the Leishmania right there. So please make sure you know the diseases they cause and which part of the world they are affecting, and so on and so forth. The next phylum is phylum Amoebozoa, which is they are the amoeba. And then Entamoeba histolytica is an organism that causes the amoebic dysentery. And dysentery means that the person, dysentery means the person has diarrhea with blood. And that's what amoebic dysentery means. Of course, it's caused by amoeba, and dysentery caused by different things, you know, different organisms, and sometimes maybe it's physiologically can cause dysentery. But anyhow, in this case, they call it amoebic dysentery. So, and the next one is Entamoeba coli, which is a commensal. A lot of people argue that is a parasite. It's not a commensal. I talked about the evolution of maybe this organism over a period of time from being a parasitic, it's becoming a commensal now. But some people call this organism called colitis. I don't know if you've heard of colitis or not. Inflammation of the large intestine, of colon, and then but your textbook and a few other textbooks say this organism is a commensal, so I'll leave it to that. And that's what I would like to ask during the exam. And then it's very hard to distinguish between Entamoeba coli and Entamoeba histolytica. You have to run some DNA, PCR analysis in order to be able to tell them apart. Entamoeba gingivalis, it's an opportunistic protozoa. 50% of the population have it, and it's transmitted through your through kissing. And this organism, if you don't keep a good hygiene of the mouth and the teeth, it can cause gingivitis, and that's what they call it, Entamoeba gingivalis. So the people who have it usually have no problem. Again, this is called opportunistic protozoa because if you don't keep a good hygiene of the mouth. The next two phylum that I'm going to talk about, they are in phylum Amoebozoa. They are not in phylum Amoebozoa. Sorry about that. They are not in the phylum Amoebozoa. They put them in a different phylum, but they are amoeba-like, and I will talk about it. They are like snail, and they have a shell, and inside of the shell is an amoeba. Okay, so I hope I'm making some sense a little bit. There is a shell, and then of course all of the slides you have in the lab on the microscopes, it's the shell of the organism. You don't actually see the amoeba. Okay, so the very first one is phylum Foraminifera, which is shelled or test. Another name for it is test. Where's my pen? Right here, it's a test. Same thing as you take sometimes a test, I take sometimes, we all take tests. And then it's a shell amoeba and made up of calcium carbonate or silicon or silicon. The other one is a phylum Radiolarians and holozoans, and it's made up of silicon. And the third one, there is a third one, is phylum Dinoflagellates, which is Serratium. And we have slide of each one of them. We have microscopic slide of Serratium, which belongs to phylum Dinoflagellates. And then the phylum Radiolarians, we have slide of it. And we have slide of 4 aminophores. Here they are, some amoeba, and here are, that's what I was talking about, radiolarians, and then foraminophorans, holozoans, and so on and so forth. These are the organisms. Excuse me. There are a few interesting organisms in phylum Amoebozoa, which are, one of them is contact lenses. It's transmitted by contact lenses, and it's called Acanthamoeba cassinelli. So the Acanthamoeba, it means spiny. So these organisms have these little spiny, if you would, that's what they call them, Acanthamoeba, these spiny structures. And then it is transmitted through contact lenses in the showers and contaminated water, usually not human to human. And then these two organisms, which I will talk about elaborately on Nuclearia fowleri in a minute, hang on. But these two organisms are called FLA, free living amoeba. That's what FLA stands for, free living amoeba, which they can cause infection. They are actually free living organisms. They're not parasitic, but they come in contact with human. They can cause devastating diseases. And of course, in case of Acanthamoeba cassinelli, it can cause blindness. It can cause it kills the cells of the cornea of the human, and eventually can cause blindness. Nuclearia fowleri has an interesting story. Naked amoebas with eruptive pseudopodia that form abruptly, and sometimes called amoeboflagellates or schizopyridins. Since the life cycle includes both amoeba and flagellata, you will see it. The name of the disease it causes is called PAM, primary amoebic meningoencephalitis. Primary amoebic meningoencephalitis, PAM. So, and then flagellated and amoeboid trophozoid, you will see the life cycle, binary fission. They divide by binary fission. You know that, we already talked about it. And transmission by amoeboid form, death within 5 to 7 days. So if somebody is infected with this organism, they have severe headache. And what happens, this organism goes to the brain and cause damage in the brain, as you heard, primary amoebic meningoencephalitis, and then the person can die within 5 to 7 days if the doctors cannot diagnose it. If the doctors can diagnose it, then great, it can be treated. So, and the treatment is by amphotericin B, or the other antibiotics they're mentioned here, miconazole, rifampicin, and so on and so forth. There is a regimen of these drugs. Here is the life cycle. When we go swimming in a body of water that is infected, so what happens? These organisms, the amoeba, we ingest the cyst stage of this organism, and then the cyst can go to our brain and become an amoeba. And then, of course, in the water, it can go between the amoeba and the flagellated organism. But when we go again in a river or lake or pond that is infested with this amoeba, we ingest it, it goes through our nose, and then voila, we come down with it. We go to a doctor, we say, "Doc, I have a headache," and what happens? The doctor says, "Oh, go home, take some aspirin." Well, really, they should test you for this. If you have severe headache and you've been swimming in a water in a nice hot days, usually you don't go swimming in ponds and lakes in cold winter days. But anyhow, then that should be an indication the doctor should put you on some antibiotics, which are listed in it. Here they are, the amoeba are infecting the cells, and they are coming out of the cells. Here are the cells that have been infected, and here is the amoeba Nuclearia fowleri. Here are some signs on the lakes and amoeboid meningitis, amoebic meningitis, and then your head above water, keep it at all times. Here is nuclear fluoride in brain cells, and here are some that they're shown. Okay, how to get ponds and lakes, I talked about that. Water in the nasal passage, immersion in bath water, and playing in warm muddy puddles after rain in hot summer days, that can do it. Placing head in troughs of water in school and playground, and in baptism it occurred, people who had full immersion. Dirty, nasty bathrooms also have been reported as transmission of these organisms. The next phylum is phylum Viridiplantae, which Viridiplantae are unicellular, colonial, multicellular green algae. That's what they are. They are pretty much green algae. You're familiar with green algae, but they are actually protozoan. And then it used to be in phylum Chlorophyta. They got rid of that classification a long time ago. Clade Plantae. Anyhow, do not worry about it. I should get rid of that one, the Clade Plantae, in a minute, because it's a protozoa. But anyhow, Chlamydomonas is the name of the organism. When the first time I started teaching biology 1 many years ago, then we had Chlamydomonas in the lab, and students looked at it, and you could see the nice green dots. You will see some pictures here in a minute. You could see the nice green dots inside the chloroplast, inside of them. Volvox, we do have slide of those in the lab, prepared slides. Here is the life cycle of Volvox and everything I want to say about Volvox. Volvox, Viridiplantae. They have cell wall division of labor. It means there are some cells responsible for metabolism, absorbing light, doing photosynthesis and making glucose molecule. And there are some cells that are responsible for reproduction. That's what I mean by division of labors. Asexual and sexual reproduction, and I will go over that briefly. Macrogametes loaded with food, and microgametes forms sperm or egg, and then zygotes multiply and break away and form a new colony. So right here, if you would, these are sperm right here, and these are egg. The sperm and egg fertilize each other. Right here is zygote, and then that undergo meiosis, and they form a colony. And remember, Volvox is a flagellated organism. Look at these. And during the exam, you get it wrong. You think they are ciliated organisms. They're not. They are flagellated. One single individual Volvox cell, it has 2 flagella. So they are flagellated, and inside of that mother colony, then daughter colonies are formed. Sometimes the daughter colonies break away from mother colony, and they grow, okay? And inside of them, another daughter colony grows. These daughter colony get out, and they grow again and again and again. So this would be the asexual cycle of this organism. The sexual cycle right here in this colony, so I don't know, in Bio 1, we talked about the viruses. They can go either on their own and multiply, destroy the cell, or they can, when the cell multiply, in case of viruses, their parts, their DNA multiply as well. But anyhow, so it's something similar to that. This colony can sometimes form a sperm, an egg, right here, sperm, an egg, sorry about that. And then, of course, what happens, these sperm and egg combine together, form the zygote, and zygote can become a colony, and so on and so forth. Okay, the next phylum, phylum Diplomonada. Mitochondria lacking mitochondria, but have mitochondrial genes in the cell nucleus, Giardia intestinalis or Giardia lamblia. Some people say they are 2 different species, and some people say no, they are one. I think they are 2 different species, but anyhow, during exam, either one you write down, it's acceptable. Here is the life cycle of Giardia, if you would. So what happens, let me write down. here is the cyst stage of Giardia, and then here is a trophozoite stage of Giardia. Trophozoite, it means feeding stage. Cyst, as we talked about it, it goes under encystment to form the cyst. Okay, I hope you know what I'm talking about. Giardia lambda trophozoites in large intestine multiply asexually by binary fission. That's what I read. Okay, so they are in our large intestine and they are multiplying and cause diarrhea. And so when we release ourselves, a whole bunch of these organisms, the cyst ones come out, okay, in the feces. Of course, there are some trophozoite in the feces too, but the cyst one are the dangerous one. And diagnostic infective cysts pass in feces. Trophozoites may be found in commonly soft and fluid. And then the method of infection in man, ingestion of infective cysts transmitted by feces, fingers, food, fomites. That's what I want you to know. And flies. Of course, flies go sit on the feces, and the cyst of this organism attached to the legs of the fly, and the fly comes and sit on our food, and we eat that food. We didn't even see the fly. We eat that food, and voila, we become infected with Giardia. That's what I mean by flies. But fomites. Fomites, how many times you put your pen, your keys in your mouth, that's a fomite. And if somebody else came and touched it that had Giardia, now you're talking about corona, nowadays, that's the route of transmission. They say, be careful, wear a mask, gloves, and so on and so forth. But that's how Giardia and many other diseases are being transmitted. Okay, so these inanimate objects, that's the definition you should know. Inanimate objects that can transmit a disease is called fomites. My mouse, sometimes I put that in my mouth. My phone, so whatever. These are all inanimate objects. If they have the parasite, the cyst on them, then it can be transmitted. Then the cyst passes in small intestine, and excistation occurs in small intestine. So we ingest these cysts, and when we ingest the cyst, it goes to small intestine. They come out of the cyst and multiply by binary fission, and multiply, and of course cause diarrhea. Okay, it does not cause dysentery, it causes diarrhea. One thing that is interesting about Giardia, I should mention right now, you guys are going to go to medical school, pharmacy school, dental school. As far as I know, all diseases are like this. In case of Giardia, they studied that if a person ingests 100 cysts, then they come down with diarrhea. If a person ingests less than 100 cysts, they do not become infected with Giardia. And that is true with most diseases. So if you ingest 1 or 2 or 10 or 20 cysts, your immune system, your body will get rid of it. But if it is 100 or more, then your body develops diarrhea. And that is true with most diseases. There is a magic number that how many you ingest or you inhale or penetrate your skin, whatever, we talk about these things later on, you come down with it. It's just not 1 or 2. That's a lot of people are under the impression that it's 1 or 2. Phylum, Parabasalia. So the organisms that I have, they have large Golgi bodies, unlike the last phylum, Trichomonas vaginalis, which you can get it by sexual contact, as the name appears. The name of the disease would be trichomoniasis. And right here, there is a diagram of it. We do have a slide of it in the lab on the microscopes. Giant amoeba, we do not have that. And then, of course, we have trichonympha in the lab. We do have it. That is right here, trichonympha. That is in the termite. In termites, the trichonympha has a mutualistic relationship with termite. So the termite eat the wood. Termite cannot digest the wood. They cannot use the wood, the glucose of the food. Trichonympha breaks down the cellulose to glucose molecule. So as a result, both Trichonympha and also the termite enjoy the single glucose molecules as a source of energy. And I talked about Trichomonas vaginalis. We do have a slide of this. And it is the only protozoa that we are studying in here that is sexually transmitted. Anyway, the signs and symptoms, maybe I should mention, it causes foul smell, vaginal discharge, and in case of male, it causes discharge, but male are less because the urethra, we talked about that, is longer. Males still can become infected with Trichomonas vaginalis, but the stats are lower. Okay, flagellate protozoa that cause African sleeping sickness. So this slide is euglenozoa and sexually transmitted diseases, Trichomonas vaginalis, Parabasalia. So they're putting it here together for you guys. But they're two different phyla. Okay. Major protozoa, taxa, phyla, phylum Dinoflagellata. Other species of Dinoflagellata, I think I talked about that. Pisidia. Yeah, it causes red tides. Other species of Dinoflagellata, we talked about Serratium. Serratium belongs, another species that belongs to Dinoflagellata. I'm sorry, my slides are a little bit out of order. Sorry about that. But has both amoeboid and flagellated form, just like Nuclearia phalarii. But it's not, they are not in the same phylum. Among 20 body types, generally feed on bacteria and algae where they can temporarily use energy stores in chloroplasts. Release short-lived toxins, that's how I said, it's called the red tide. And then you should not eat the shellfish from this area by red tide. If you do, it can cause death. Here they're showing you red tide, as I mentioned it. So that's the organism that causes this, and then because of the toxins they produce, and then we should stay away from the food that is in this area. Okay, Serratium, as I talked about it, these are some of the organisms that we talked about. Finally, the last phylum is phylum Apicomplexa. I put a picture of a tank in there for there. All of the members of this phylum are parasitic. They are parasites, and they have a structure called apical complex. And two of the structures in the apical complex, there are more. I only mentioned two of them, rope trees and micronemes, because these two structures allowed the organisms to get inside of inside of the host cell. So example, this is the host cell, for example, and then this is the Protista. They release enzymes and molecules to digest away the cell membrane so the Protista will get in, okay? And when they get in, these organisms do not have flagella. They do not have amoeboid pseudopod, I should say pseudopod. They do not have flagella, they do not have pseudopod, they do not have any locomotion structure, but one way they do move is called gliding. Gliding. And what is gliding? If the organism like this, and in Bio 1, we talked about it, your cytoskeleton, there are microtubules underneath, and the cell membrane move over the microtubules, just like wheels of a tank. So imagine this would be the cell membrane, and this would be the microtubules. Okay, I don't know in Army, a movement of tank, they call it what? But in Apicomplexa, that type of movement is called gliding. Okay, anyhow. So pseudopod in some intracellular stages, you will see that flagella in gametes of some species, asexual and sexual cycles, some requires intermediate hosts, and some develop oocysts. Here they are, or oocysts, or spores. We'll talk about all of those. But here is the apical complex structure, and here are some of the structures listed for you in here, but only expect you to know 2 of them, rope trees and macronemes. And then they have other structures. They have other structures that are like mitochondria, they have Golgi apparatus, nucleus, and rough endoplasmic reticulum, and so on and so forth. And these are the cysts, oocysts, sorry. They are oocysts. When the sexual reproduction occurs, they form the oocyst. The first class that you should know is class Coccidia. So not every phylum, I gave you name of a class. Apicomplexa, I gave you one, and then Euglenozoa, I gave you one also, or 2. I gave you 2. Euglenozoa, I gave you 2. Euglenidia, and then Trypanosoma. So Coccidia, most important class because the members of this class cause disease. They cause death on planet Earth. So pyroplasma in cattle, it causes Texas red water fever caused by Babesia bigemina, and then pyroplasma is the name of a class, but in cattle, the name of the organism that causes disease. We human get Babesia as well. When we human get Babesia, some doctors, some clinicians, they on the microscope, when they look at it, they think it's malaria. So it can be easily be mistaken by malaria. Babesia is easy to treat. Malaria is not that easy to treat. Okay, so, but 2 different species. The other organisms in this phylum is Cryptosporidium, cause severe diarrhea, not dysentery, but it's a diarrhea that you cannot get rid of it as easy, okay? You probably need hospitalization. But Giardia, your body gets rid of it in 2-3 days. And a lot of people in other countries, they have Giardia and their body got used to it and they live a normal life. Not they did not get used to diarrhea, their body goes back to normal and they have normal defecations, but they just have Giardia. And they don't even know it. In the United States, I say is, in all my lectures, in the United States, our gut is sterile. We do not have any parasite in there. Other countries, their gut is 1 or 2 or 3 or 4 parasites they have. So Cryptosporidium, there were 2 outbreaks in Washington, D.C. area and Milwaukee area, and then a lot of people were hospitalized. There are literature out there. So anyhow, Coccidia again, intracellular, Eimeria and Isospora cause coccidiosis in chicken farms and poultry farms, turkey and chicken. There are horrible, I've heard of horrible stories. You know, the farmers get up next day and everything is dead on the farm. Isospora in AIDS patient can cause a problem. Eimeria tenali, again in chicken, in poultry farms, turkey and chicken can cause a lot of problem. Okay, so I think I gave you a rain check on schizogeny and sporogeny. So let's come back to it. So in the chicken, let's talk about Eimeria in chicken a little bit. So schizogeny, they form a gametes, right? The microgamete and macrogametes, they come together, these gametes, microgamete and macrogamete, they are combined together, called fertilization. And then after fertilization, then the oocyst is formed. So they do not like to give the term zygote to these guys. They give the term to these organisms, they give the term oocyst, not a cyst. So this would be a nice question, essay question. What are the differences between oocyst and cyst? And give organisms that explain each one of them. So anyhow, the oocyst then is ingested by a chick, by a chicken. When oocyst is ingested by a chick or chicken, then what happens? It goes to the large intestine and multiply and multiply and multiply. And the name of the process is called schizogeny in the large intestine. And then the oocyst, when it's multiplying, inside of the oocyst, you have, of course, the zygote. Look, it's one nucleus, but inside of the oocyst, this is the oocyst, right? Inside of it, so you have then one nucleus become two, two becomes four, and that stops. Most species are like that. They stop at four. I'm talking, again, you're talking about Apicomplexa. Okay, so it stops at four, and then when another chick picks up these oocysts, and these four go under existment, and they go, so now you have four, and they go find an epithelial cells, columnar epithelial cells, and they go inside and multiply and multiply. The nucleus multiplies, but they don't form a cell membrane around themselves. After the nucleus multiply, which is called schizogeny, then they form cell membrane around themselves. I hope I'm making some sense. And that process is called schizogeny. This process right here is called sporogeny. This process right here is called schizogeny. Okay, so 8 sporozoites in case of Amir Bio. In case of Cryptosporidium, there are 4 sporozoites inside of it. Toxoplasma gondii, there are 8, I'm sorry, there are 8 sporozoites. I'm forgetting these things over the years. But anyhow, and they found a new host, and we talked about it. Toxoplasma gondii, I will talk about it next lecture, but it's devastating in AIDS patients. Half of us in this world have it in the United States. Let's go. Half of us in the United States, half of the population in the United States have Toxoplasma gondii, and we don't even know. Definitive host is cat. It means sexual reproduction occurs only and only in cat, wild cat, domestic cat, and we'll talk about that a little bit. Possible intermediate host, which is us. Plasmodium is malaria. So malaria transmitted by mosquito. Anopheles, you probably have heard of that. You already know that. Sporozoites injected to human by insects, which is, I already talked about that. Sporozoites in liver initiates schizogeny, and plasmodium, already spelled out plasmodium. So I can write down P. falciparum. Plasmodium falciparum is the most devastating of the 4 malaria species that we human get. Okay, so Plasmodium falciparum, it causes death, and it penetrates red blood cells after 1 generation in liver. In red blood cells, trophozoites feed on hemoglobin, sexual reproductions, macrogamy, and mosquito ingests gametes and fertilization oocyst right here. That's what I want to talk about. Okay. So the mosquito, Anopheles, female, it has to be female mosquito. The male mosquito do not feed on human, do not feed on blood. Okay, just the nectars of the trees and so on and so forth. But female mosquitoes on both. Anyhow, they come and ingest our blood, and in their feces or saliva, they release their injected sporozoites right here. Okay, and these sporozoites go to the liver. In the liver, schizogeny occurs. Look, in the liver cell, this is the liver cells, the nucleus divides and divide of plasmodium. And then they form a cell membrane right here. Okay, they form a cell membrane. They break out of liver cells, the hepatocytes. Of course, they destroy the hepatocytes. And they call them merozoites. The merozoites enter red blood cells. Okay, in the red blood cells, again, they multiply and multiply, and they form microgamete and macrogamete. Okay. Then the mosquito comes and take a blood meal. Female mosquito ingest the gametocytes, the microgamete and macrogamete, and inside of the gut of the mosquito, the microgamete and macrogamete come together and form the oocyst in the gut of the mosquito. In the gut of mosquito, as I talked about its sporogeny, you have one, this is one nucleus is under this, it goes and multiply and multiply and multiply. Of course, this is sporogeny. The name of the process is sporogeny. Okay, and then what happens, burst open, then you have these merozoites right here. They come to the salivary glands, and then when they take a blood meal from human, from the salivary glands, they come out these merozoites and then sporozoites and inject human. So what it is, the mosquito is the final host, and a human is intermediate host. Final host or definitive host, same thing. Final host, definitive host, same thing. Okay, and human is intermediate host, where asexual reproduction occurs. Sexual reproduction occurs in mosquito. We can stop this by destroying the mosquito. It happened in the United States. When the settlers came here, there was malaria, but by 1920s and 30s, United States was successful to eradicate these mosquitoes from US. So there is no malaria in US. But there was. These are the cysts in the mosquito of the gut of the mosquito. Slide of the cyst I'm showing you. And then, of course, you know, malaria is a problem all over the world. Africa, South America, Asia, you name it. But Western Europe, Australia, these are the countries have been able to control it. Most of the industrialized countries have been able to control it, but it's a problem the rest of the world. Cryptosporidium parvum, I talked about that a little bit, live in small intestine and respiratory epithelium, direct life cycle. Oh, the life cycle of, for example, the one thing I did not mention of malaria is not direct, it is indirect of malaria. Life cycle of trypanosoma is indirect. Life cycle of leishmania, they all need another host. So it's indirect life cycle. Giardia is direct life cycle. Cryptosporidium, direct life cycle. You don't need another arthropod or something else. Directly from human to human, of course, through feces, through fomites. It doesn't need a mosquito. Okay, so I hope I'm making some sense. So the oocyst about 4 or 5 micrometer, very small. Microgametes and macrogametes are formed. The sexual reproduction, oocysts in soil and water, it can become airborne, and outbreaks in Washington, D.C. and Milwaukee, which we talked about. Yeah. Oh, I'm sorry, one more phylum. Phylum Ciliophora, I thought Apicomplexa is the last one. Mostly free living, some parasitic, some commensalism and some colonial. Some sessile, like Volvox was a colonial as a colony. They form as a colony. I hope you know what I mean. Some sessiles, like Sartorians, are sessile and larger than most protozoa. If you look at them on a microscope, Paramecium is bigger than the rest of the protozoa you have seen. Multinucleated, they have 2 nuclei, macronucleus and micronucleus. under undulating membrane, cilia are fused into a sheath, cytostome, the mouth, oral groove, two contractile vacuole system. Already talked about that. Passage of water and food in paramecium is oral groove to cytostome to cytopharynx to food vacuoles, indigestible out by cytoplasm. Okay, they do multiply by binary fission. Micronucleus divide mitotically and macronucleus elongates and divide amitotically. Conjugation, when they come together and exchange nuclear material, and autogamy, it means self-fertilization, like Balantidium coli. They do that. Balantidium coli is the only ciliated organism you should know that causes diarrhea. It causes disease. Okay, and then we have another ciliated organism in the lab, which is Paramecium, and that does not cause any disease. Okay, Balantidium coli causes diarrhea. Here is, I already talked about contractile vacuoles, so I'm not going to talk about it a little bit. Here is the oral groove right here, and then this is the cytostome, the mouth of the animal. This is called cytopharynx, a little bit down here. And then these are the food vacuoles they form when the food comes this way. And they digest it. You learn that in Bio 1 with lysosome and so on and so forth. The indigestible get out through cytopage or cytoproct. Okay, so macronucleus, the function of macronucleus is for metabolism, and the function of micronucleus is for reproduction. Macronucleus metabolism, you learn that in Bio 1. All of that in us is metabolism and reproduction in one place, because DNA is responsible for protein synthesis, right? We talked about that. So that's what I mean by metabolism. Here are some pictures of fun pictures of gullet, another for oral groove, macronucleus, micronucleus, contractile vacuole, food vacuoles, and trichocytes. Trichocytes, I think you learned it in Bio 1 or not, I don't know, but at the base of the cilia, these are cilia, at the base of them, there is a structure that holds the cilia in place, and that is called trichocyte. Okay, this is my drawing from the beginning. I told the people do not get rid of it, but here is a student make this nice and beautiful PowerPoint. So this is conjugation. When two paramecium come together, this is macronucleus, this is micronucleus, right? This is micro, this is macro, okay? So they come together, what happens? Micronucleus undergo meiosis 1 and then meiosis 2, each cell. Then everything disappears. Even the macronucleus disappears. Micronucleus disappears. All of the other micronuclei disappears. And then what happens to the leftover, this is the leftover micronuclei, which undergo 2 stages of meiosis. They undergo mitosis. So this is mitosis. It's not macronucleus. Maybe I should have drawn it a different color, not red. Okay. And then they exchange nuclei, micronuclei. One goes to another cell, and this one goes to another cell. And of course, that is called autogamy. Then macronucleus appears. Of course, this is a zygote. The 2 separate, and then each one of them undergo binary fission. So, having said that, when organisms exchange nuclear material, like human, like most species on planet Earth, what happens? The genetic variability increases. When the genetic variability increases, then natural selection, if a disease comes, since we are genetically different, we can wipe out some, but some survive. in a population. Some dies, but some survive. The ones that survive can multiply again, and then that disease will not destroy them. I think at the end of the semester, we'll talk about evolution a little bit, and this will come to picture. So sexual reproduction causes genetic variability. Genetic variability causes the survival of the species. If all of the species were genetically alike, then if a disease like HIV or corona, we are dealing with it right now, would come, it would wipe out the whole population. But that's not the case. We are all genetically different, so it will wipe out some portion of the population, but the rest of the population survive and they can multiply. Okay, this is not a complete table, but you complete it. It has the name of the organisms: Trypanosoma cruzi, Trypanosoma brucei, Leishmania, Giardia, Trichomonas vaginalis, and Amoeboid wine, Amoeba histolytica. And then sporozoans, do not go, that's an old name, sporozoans, Plasmodium, Toxoplasma gondii, and ciliated one, Balantidium coli. Most all of these, you have picture of them, except you have microscopic slide of them, except, yeah, Entamoeba histolytica, you have Entamoeba coli in the lab. Make sure you know where they are found. All of these you should know. What disease they cause, the name of the disease: Chagas disease, sleeping sickness, leishmaniasis, giardiasis, amoeboid dysentery, so malaria. So that's the name of the disease. And then of course the signs and symptoms. These are signs and symptoms. Make sure you know the signs and symptoms, okay? And then how they are being transmitted. Make sure you know all of, I talked about more diseases. For example, Naegleria farae is not here, Cryptosporidium is not here. Oh, I talked about more diseases than what is in this diagram. What you can do, you can complete this table based on lecture notes, and then we go from there. I think that's the end of it. That's the end of the material for, I still, there's a Toxoplasma gondii lecture left, and I will talk about that, and that would be the end of the material for exam number 2, after Toxoplasma gondii. Okay, so see you until Toxoplasma.